For those of you who don't know who I am a consultant ophthalmologist. I am based at Moorfields Eye Hospital in London and Great Ormond Street Hospital for Children. I specialize in genetic eye disease and inherited retinal diseases, and I see children and adults with these conditions. I'm also a professor of molecular ophthalmology, and I have research labs based at University College London and the Francis Crick Institute in London. And I undertake a lot of research into these genetic eye diseases. But a specific research focus of mine for many years has been choroideremia. Since probably even before 2011, I worked on choroideremia during my PhD, which was between 2005 and 2008. So it's a massive honor for me to be able to come to give you this talk today and to continue to support patients with choroideremia, their families, and the Choroideremia Research Foundation. So to begin with, we're going to start right at the basics, and we're going to ask ourselves about the structure of the eye so that you all know what I'm talking about when we get into slightly more detail. So I have a schematic on the screen which shows a cross-section through the eyeball, and it highlights the various structures from the front of the eye, the clear part where light enters, the cornea, the anterior segment where we've got aqueous humor in the front that sits in front of the iris, which then lays itself upon the lens. And there is the round gap, which allows light through the center of the iris, through the lens, which is called the pupil. And then you've got the tough white outer coat, which is called the sclera. And then the layer that is lining the back of the eye is called the retina and the choroid. And so when light enters the eye, it first hits the retina. And the retina is composed of several different cell types. These are essentially neurons that are connected to one another that send signals to the nerve, the optic nerve, which then travels to the brain, to our visual cortex, sending signals about what we're seeing, and formulates that into the images that surround us. Behind what we call the retina, the light sensitive layer, there's actually a single cell layer that is almost like the mother cells of the retina. It does the housekeeping and looks after them, and that's called the retinal pigment epithelium. And behind that is a layer called the choroid, and the choroid is actually formed of 3 or 4 layers as well, but it makes up the blood supply to the back of the eye. So that's the basics in terms of the structure of the eye. Now, within the retina, you have light sensitive cells, and you have 2 types. You have rods and you have cones. Your rods are what help you to see in dim light and your field of vision. And these are situated in the peripheral part of your retina. And I'll be showing you pictures of that in a minute. And then you've got your cones, and your cones help you to see fine detail, color, faces, read print, and they're located in the central part of your retina called the macular area. So I'm going to just go to the next slide. Now, this is a photograph of the back of a healthy eye. Now, I don't know if you can see the cursor, and for those of you, I'm sorry if you can't see this screen, but I'm just going to verbally describe what I'm seeing for you. So there is it almost looks like a big orange sun. I remember the first time that I looked into the eye through the operating microscope, and I almost felt it looked like flying above the Nazca lines in Peru. It was just amazing. It was like this kind of red earth with these lines that were all over it. But what we see here in this image is this kind of brighter yellow circle, which is your optic nerve. So that is the nerve that carries signals from the retina to the brain. And then you have all these red lines coming out from the optic nerve, spreading across the retina. These are your blood vessels. So these supply the retina with oxygen and glucose and nutrients and carries the deoxygenated blood away. So your arteries and your veins. Then right here in the center, if you can imagine light coming in through the eye and it hits that central part at the back of the eye, it's a slightly deeper orange in color. That's actually your fovea, your area of central vision, and surrounding it is your macula. And this is the area where it has the most highest concentration of your cones, your area of central, fine detailed vision. And then all around the rest of the retina, where you have this sort of orange color, are your rods. Your rods, as I said, are what help you to see in dim light and your field of vision. Next to this is a histological section of a human eye, and it just shows all the different layers of the retina. And I'm not going to, go into too much detail, but just to say that right at the bottom, if we start at the bottom, that's your choroid, your blood supply to the retina. Then you've got a single cell layer, which is called the retinal pigment epithelium. There, remember, your housekeeping cells for your light sensitive cells, which are your photoreceptors that sit above it. And then you've got various different layers which correspond to the neurons and the connections that send those light signals that have been converted into chemical signals back up towards the nerve that then goes and is passed on to the brain. I suppose I'm going to pause there. Are there any questions on the anatomy at the moment? And when I talk about rods and cones and the retinal pigment epithelium in the choroid?
No, not yet.
Okay, good. So we're going to start now moving into choroideremia. And the next question is, what causes choroideremia? Well, as you know, it's a genetic condition. So choroideremia arises from a change in your genetic code. And specifically, it's a change in a gene called the CHM gene. Now, to put things into context, so our genetic code, every single cell of our body has a copy of our genetic code. And our genetic code is around 3 billion letters in length. And I always say to people that if you were to ask a secretary to type out 3 billion letters of your genetic code, they would introduce a number of typos. So all of us carry around 4 to 5 million typos in our genetic code, spelling mistakes. Of that number, well, how should I put this? Those typos make us unique. So I have 5 million typos, someone else has 5 million typos. It makes us who we are. But of that 5 million typos, 10,000 of those changes can potentially be disease causing. And that's not something you can control. So if there are mothers in the audience, patients who've been affected by this, I know it is an inherited condition, but where it started and how it started, that's not your fault. That was because of natural variation and a spelling mistake in a gene, and it happened to be in a gene called the CHM gene, which causes choroideremia. Now, again, of that 3 million letters of our genetic code, we have 20,000 genes. And one of those genes is the CHM gene. And if you happen to have a spelling mistake in that gene, that's what leads to the condition. Now, when I talk about these kind of spelling mistakes, what do I actually mean? Because there's lots of different types of genetic mutation. So some patients can just have a single letter change, a single letter of your DNA could change. And I've given an example here. Instead of the spelling CAT for cat, the T could change to a G, and instead it makes CAG. Now, if I say to you cat, you initially straight away think of a little furry animal with ears and a tail, cute little pet. And when I say the word cag, that doesn't make sense at all. That's kind of what's happening in your gene. You're getting a letter change that is changing the whole meaning. So it doesn't make sense anymore. And therefore, you can't make the correct protein. The protein doesn't know, it doesn't make sense, it doesn't work properly. And that's what leads to the condition, the lack of that protein. Those kind of mistakes where it's a single letter change, they may be called nonsense mutations, missense mutations, and I'll go into a bit of that in later on. Sometimes instead of a single letter changing, a letter might just get deleted. So again, if we take cat and we deleted the A, it would just shorten to CT. Well, that doesn't make sense either. So that's therefore caused the same issue with the protein. And then sometimes an extra letter is inserted. So an extra G could be inserted. So again, you're losing the meaning of cat and you've got something called cagta. Any of these kind of genetic changes can lead to the condition. Sometimes it's not just single letters being added or changed. Sometimes it can be hundreds of letters. Sometimes the whole choroideremia gene can actually just be deleted. Sometimes the gene could be turned back to front. So there are lots and lots of genetic mutations that can occur that can affect this gene. So the CHM gene provides the instructions for making a particular protein, and that protein is called Rab-Escort protein 1 or Rep1. And Rep1, the protein, is actually found in every single cell of our bodies. Humans have a similar gene to Rep1. It's called Rep2, and it can actually compensate for the lack of the Rep1 if it's not working in basically all other tissues of the body except for the retina. And that's why we see our sight loss condition affecting our retina. I've got a picture on this slide which essentially shows DNA, your genetic code, that 3 billion letters. There's a subunit in that genetic code called a gene. And if we can imagine the CHM gene, in order for that gene to produce a protein, it needs to produce a set of instructions, which is called mRNA or RNA. And then our protein making machinery reads that RNA and it then goes on to produce the protein. So this is what's happening within our cells. And what can happen is if you have a mutation in the CHM gene, you still can produce the mRNA, the messenger RNA, the instructions, but the instructions can have the mutation in it, so it doesn't make sense. And so when your protein making machinery go to read it, it doesn't form a correct protein. And in some cases, it just stops. It doesn't bother making the protein, or it makes a protein which degrades very quickly. Any questions at this point?
Nope, not yet.
Okay, good.
You're doing a great job. This is awesome. Thank you.
Okay, good. So then how does this actually affect the retina? So the Rab-Escort protein essentially works in shuttling specific proteins inside retinal cells. What they also do is kind of modify them so they can be activated. And so when the Rep1 protein is either not present because of that genetic mutation, or it's just not working, it can't shuttle these specific proteins where they need to go, and it can't activate them. And because of it, because it's basically not working as it should, the cells cannot function properly, and therefore they eventually die off. And that's what leads to the retinal degeneration. And when those cells die, that's when you start to lose your sight. Now, in terms of choroideremia, we know that the first types of cell that are affected in the retina are the rods. The rods are the cells that help you to see in dim light and your field of vision. And that's why in our males, in our young boys, they often will complain of difficulty with their night vision. After that, then they discover that they're having problems with their field of vision because it's narrowing. And then eventually, once the cones start to become affected, that's when they start to, for example, lose their contrast or their color vision and eventually their central vision. And because this is a genetic change, this can be passed on to future generations. And I'm going to talk about inheritance. So this condition is what we call X-linked recessive. We know overall it affects between 1 in 50,000 to 100,000 males, and males are much more severely affected. Females can be carriers, but they can also develop symptoms. And if you have, for example, a mother who is a carrier of the genetic mutation. Now, when I say women are carriers, we women have 2 X chromosomes. And the way it works is we have a healthy X and an X that has this mutation in the CHM gene. And so when we go to have our children, we have a 50-50 chance of passing our healthy X or our mutated X to our children. If, for example, our partner gives his X chromosome, then we will have a daughter, but that daughter will have a 50% chance of either inheriting the mother's healthy X or the mutated X. So a 50% chance of being healthy or a 50% chance of being a carrier. If the partner, the male, passes his Y chromosome, then the mother, again, it's very dependent on the mother. If she passes her healthy X, then the son will be unaffected. But if she passes her mutated X, then the son will be affected. And there she has a 50% chance of doing that. So 50% chance of having an affected son or 50% chance of having a healthy son. Now, it doesn't always come from the mother. It could actually just happen in the child when they're a ball of cells developing in the womb. That's called a de novo sporadic mutation, where, for example, a male fetus, or when it's a ball of cells, develops a mutation in the CHM gene, and then that individual, when he's born, he then starts to develop choroideremia. But then that individual will then pass it on to future generations. So that's how it starts. It starts as what we call a sporadic mutation, but then is propagated as people have children and further kind of generations. I wanted to talk a bit about female carriers, in that most female carriers are essentially fine. They don't show any signs of the condition. Maybe in later life, they may complain of a bit of night blindness. And remember, that's because women have 2 X's. But if women walked around with 2 X's all the time, they would be sort of like super women, right? So our cells don't allow that. What happens is 1 X is always switched off in our cells. So only 1 copy of our X is working. Now, that choice of which X is switched off or not is random. it's a kind of toss of a coin, 50-50. Is it going to be the healthy X or is it going to be the mutated X, for example? And so that process is called X inactivation or X lionization. Means the same thing. One X is being switched off randomly to reduce the double X action. And so there are sometimes some female carriers where more of the healthy copy of the X is switched off, and therefore more of the unhealthy mutated X is actually working, but because it's got a mutation in it, it's actually not working very well. And so those female carriers can show more signs of the disease. Equally, there can be some female carriers where more of the unhealthy X is switched off, and so they have more healthy copy of their X working. Now, just the other thing I wanted to note here, which a lot of you will be aware of, but nowadays, when you get a molecular diagnosis where you have your genetic result confirmed and you want to go on and have more children, obviously I've given you the kind of risks, and you can see that in this image here of your chance, for example, if you're a female carrier, of having an affected son or a carrier daughter. But if you're an affected male and you're going to have a child, so you have no choice but to pass your affected X to if you're going to have a female daughter. So your daughter would be a carrier. But if you were to have a son, they would not get the disease because they have your healthy Y copy. Okay. Now we have something called pre-implantation genetic diagnosis available. And that's essentially a form of in vitro fertilization, or IVF, where you can take an individual's sperm and their eggs and you fertilize it in a dish. And when they're a ball of cells, you can take a cell and you can check it to see if it's carrying a mutation in the CHM gene, and then only implant the healthy embryos back into the womb. And that is a service, at least in the UK, it's available on our National Health Service for free for individuals, and I'm sure that it's available, across the world now. But that is a form of family planning that patients with choroideremia can now access, and parents who are worried about having further children with this condition. Okay, any questions on that? It's quite hardcore genetics there.
There's no questions yet, but as a female carrier, an older female carrier, some of the things that you've shared today, I've not heard before. So it's quite astounding. So thank you for taking the time, and thank you for the slow process by which you're sharing this, because so many of us are interested in that very topic. So no questions yet, but just thank you and carry on.
Okay. All right. So I'm going to talk about patient symptoms now. A lot of what I'm saying, because I can't remember what the next slide is, so we're going with the flow. Some things will be repeated as well. So in terms of an individual's symptoms, so this can actually vary quite a lot between individuals. I did mention that the first symptom usually is a symptom of difficulty seeing in the dark. That can be noted in early childhood. I say early childhood, it could be noted in teenage years. It might be later than that. Some people just don't even pick up on it. Some people go straight to, oh, I was actually driving and I had a car accident, and that's when I realized that actually I couldn't see very well in my field of vision, and they never knew they had night blindness. But just to say that would be the first symptom, and that's because it is our rods that are degenerating first. And so when your rods are not working, that's when you struggle in the dark. I get a lot of patients who say that they only noticed it in their late teenage years, when they used to go to the cinema with friends, and they couldn't see, they couldn't orientate themselves, they were tripping over steps, whereas their friends seemed to be totally fine. Following that, it's usually a sort of patchy loss in your peripheral field of vision, because that's how choroideremia works. You sort of get these patches of degeneration that sort of grow bigger and then kind of coalesce, but it's your peripheral vision that will go first. And then you'll be left with pretty good central vision for a number of decades. And then you may start to notice loss in your contrast sensitivity, being able to discriminate between kind of shades like navy blue and black and dark brown, and then your color vision might be affected. You can often find it quite difficult to transition from being outdoors in bright light and coming indoors. That can take quite a long time. And then eventually it will affect your central vision as well. So what care should you receive? Well, if I can emphasize anything, I know it's a small group on this call, but, spread the word. The most important thing, in my opinion, the one test you can get done is genetic testing. I'm sure all of you have had this done. You all know it's choroideremia, but just to say, if there's some older people on this call, 10, 15 years ago, people would look into the eyes of choroideremia patients, and it was an exam question for us. They would say, spot diagnosis, what do you think that is? And we would look in the back of the eyes and go, yeah, that's definitely choroideremia. Because you see, as you can see in these pictures, that the retina, the retinal pigment epithelium, and the choroid degenerate. And so when you look into the eyes, when you see that yellow hue shining back at you, that's actually the white of the eye, your sclera that you're seeing. And that was very what we call characteristic of choroideremia. Now, in the images I've got in this picture, I would have definitely looked at both, not the healthy picture with the nice orange glow, but the two kind of pale yellow pictures. I would have looked at those and gone, yeah, definitely choroideremia. My professors would have said, definitely choroideremia. So we've got one image of a 40-year-old male who's suffering from night blindness and peripheral field loss. The other one is a 68-year-old male, again, suffering from night blindness, visual field defects, but also central vision loss. And that's understandable because he's, 18, maybe no, 28 years older. So he's at kind of end stage disease. We actually undertook genetic testing for these individuals. We found that the 40-year-old male indeed had a choroideremia, a mutation in the choroideremia gene, the CHM gene. But the 68-year-old, we couldn't find a mutation in the CHM gene. So we're kind of scratching our heads, but this looks just like choroideremia. And then we took a blood sample and we checked to see if he had the choroideremia protein, the Rab escort protein 1, and that was normal. And so we're like, oh, this is not choroideremia then. We then did whole genome sequencing on this patient and found he had mutations in the RPE65 gene. So what we now call this in our field is a phenocopy. This is a patient that looks like a choroideremia patient, but has a genetic mutation from a different gene. And actually, now, if this 68-year-old presented when he was, say, 30, and he had RPE65 mutations, he could actually access Luxturna. So it makes a massive difference in terms of your management going forward. So please get genetic testing if you haven't already, and spread the word to those who haven't. So this is now pictures of some female carriers. And as I mentioned, a lot of female carriers are not affected at all, but it can be variable. I've got a picture of a 35-year-old female who is suffering from night blindness, and her father had RP. Now, also be warned of a diagnosis of RP. Again, 30 years ago, pretty much anyone who had night blindness and field defects were told they had RP. It was a sort of an umbrella term, but it wasn't RP at all. The father had choroideremia, and she is now a female carrier. That's why she's now started to suffer from a few of those symptoms. Here on the other side is a really pale retina looks like a male patient with choroideremia because it's lost so much of the retina and the choroid. This is actually a 40-year-old female who is actually quite severely affected by the choroideremia mutation, because more of her healthy X has been switched off and her unhealthy X has been left switched on, but it's not got the choroideremia gene working. So what care should you receive? Well, you should be having annual visits with your ophthalmologist. If you can do any tests, because I know there'll be people from around the world here, and, you know, access to healthcare is not always equitable amongst all countries. Sometimes you have to pay for your investigations and your care. So the two tests that I believe are the most significant, if you have once a year, would be an OCT, which is basically, it uses infrared light to take an optical cross-section of the back of your eye, and that's the middle pictures here. It allows us to look at the kind of boundaries of healthy and unhealthy retina and how it changes over time. And the other imaging test is something called a fundus autofluorescence, where a lot of you will know you get a really bright blue light shine in your eye, and we get to kind of see how much of the healthy retina is there and how much has been lost, because it looks at those retinal pigment epithelial cells and sees how many are still kind of alive, stressed, or have degenerated. Obviously, you will also have your visual acuities done. Some people do have visual fields tests done annually. That's a little bit more variable.